Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12th Molecular Basis of Inheritance Part 10. In the interviewing the questions, how do the RNA polymerase able to the catalyst all three steps? How do they get they participate in the process of the total transcription process, which is the initiation process, elongation process, and then termination process. They are having how many steps? Three steps as present: initiation, elongation, and then termination. Now, here, see RNA polymerase. Here, mostly in this RNA polymerase is only capable as in that catalyzing the process of the elongation. So, only one enzyme they have any capacity. The total two as this process. That enzyme is what? RNA polymerase. DNA dependent RNA polymerase they have any capacity to get completely to get do this entire the initiation, elongation, and then the termination process. The association with the transiently the initiation factor, that is in a complete word sigma factor. Okay, now in the sigma factor, in this in the sigma factor and the terminator factor is rho. It is in the initiated to as in the terminate transcriptional unit. So before once we observed in this total, say in this total factors here, the RNA polymerase they are also capable of having the catalyzing of the process of elongation. The associated with the translation the initiation factor. This is what this is in a, that is first one is what the sigma factor. This is the first one is what sigma factor, and the termination factor is what this is in a rho factor. This is what in a rho factor initiate as in a they completely terminate in the transcription respectively. So here the first sigma factor in that when a sigma factor they initiate to get forms as what in a, that is in a rho factor, that is in a rho factor. Let us the association with us in these factors they are alter the specifically the specificity of the RNA polymerase either initiate as a terminate. So in this enzyme they get participate the either in the initiation process, either in the elongation process or in termination process. Okay, this is. And after when you observe as in some bacteria, in that in some bacteria, since the mRNA does not they require for any processing of become in a active, since the transcription and translation takes place in the same compartments. Here in some bacteria, for example this is in a bacteria, in this bacteria they no require for as in that to both the transcription and translation, both in a, their transcription and then translation, and then here translation they are no required, because why? This is the one compartment, the both the transcription takes place here and then translation also takes place in only one compartment. So there is no separation of the cytoplasm nucleus in the bacteria. So because here they are having as a complete nucleus is present and then completely the cytoplasm is present. Okay, the many times the translation can be there begin the much before the RNA fully transcribed. So that purpose, that is the reason. So that purpose what happens is totally complete there easily to get transcribed, and that completely subsequently the transcription and translation can be the coupled in a bacteria. So in the bacteria both in the transcription and the translation only they look like as an a coupled. Okay, but once you compare to as an eukaryotes, when you compare to as an a here eukaryotes not like in eukaryotes, not like. Why? See here, mostly in the eukaryotes there are two types of additional complexities will be present. In the eukaryotics they are having as in a completely there are two kinds of the compartments is present. Let's see here the first one, there or at least the three RNA polymerase the nucleus they require the RNA polymerase found in the organelles. So in all eukaryotic cells the completely the three types of RNA polymerases they get participates there, the clear cut division of the labels. So these are completely called as a clear cut of the labels. Here RNA polymerase one transcripts as an or RNA. So for example they having a first one is what, that is an rRNA, that is what first one is what rRNA, first one is rRNA. The first one is rRNA. And Here this rRNA that contain as in the 20s yes and 18s and 5.s their ribosomes as present, whereas in a RNA polymerase 3 that transcribed by as in a the transcription of in a tRNA they contain as in a there's 5 small ribosomal RNA and small nuclear RNAs means the small nuclear RNAs as present. Which one? That is in a RNA polymerase 2, that is in a tRNA. And last one is what? That is in RNA polymerase 2, that is in a mRNA. That is called as what? Heterogeneous nuclear RNA. That is in a heterogeneous nuclear RNA. Okay, now next one, let's see here in the second complexity. The second complexity is what? The second complexity that has in a primary transcription contains in a both exons and introns. They are as in a non-functional. Hence, they are subjected to as a process as called as a splicing, whereas an introns are removed and exons are joined, defined as an orderly. So here before once we observed, see in this process these are completely in that region they are the coding sequence. These are exons. So exons to get joined and introns are to completely they are removed. Okay, now in this process only guanosine, that is a triphosphate. Okay, that is next. Let's see. HNRNA the undergoes the additional process of called as a capping and tailing. So what is the capping tailing? So in this capping, the usual nucleotides in the methyl guanosine triphosphate, the addition of in a 5 end. So for example, when you take as an A, it is in A1 template stand. So for example, it is in a 5 prime, it is in a 3 prime. So here they are having as mostly has what? That is in a guanosine triphosphate. They contain as what? In a, that is in a guanosine triphosphate. The 5-methyl guanosine triphosphate is present in that region. In this region, they are having as in a the methyl guanosine triphosphate. Methyl guanosine triphosphate is they are present in that HNRNA. That is called as what? In a capping process. In this tailing process, what adenylate means, see here in the 3 prime region, here has in a around as 200 to as in a the 300 there the complete amino acid residuals as present in that region has in a 200 to 300 amino acid residuals as present in the 3 end. The template the independent manner. So independent manner when they are having as in a capping, when they are having as in a capping and then their tailing region, the total DNA will they get fully, yeah, they given it is in their fully they proceed HNRNA. Then what happened? You get as in a completely the heteronuclear RNA is to get formed. That is now we call as an mRNA. The transcription the transported out of the in a nucleus, that translation will be they get starts. Okay, for significance of such as a complexity is now the beginning to be there understood. So when the before what happened and after what they going on, we clearly get sequence. Now the split genes, the arrangement of respect to the probability in a ancient features of the genome. So this information based on their previous information also we get easily their uptake. The presence of the introns, they are reminiscent of in a antiquity, and the process of in a their splicing represents as in a dominance of in a RNA world. So in this total the splicing or maturation of mRNA, this total is a part of dominance of RNA world. In these recent times, the understanding RNA and RNA dependent RNA polymerase in a living system has in the assembled more important. So here understanding of RNA and RNA dependent polymerase, this total they given as in more importance. Okay, this is in a different types of in a RNA polymerase exist in a eukaryotic cells. Remaining to discuss in next class. Thank you for watching. Please share, subscribe and like.